Atomic absorption spectroscopy is an instrument method of quantitative and qualitative analysis. Quantitative analysis refers to determining the amount of analyte in the sample, while qualitative analysis refers to determining whether an analyte is present in the sample. Atomic absorption spectroscopy is used to identify elements in the wide variety of substances. It also uses the characteristics absorption spectrum associated with each element. It is a very common technique for detecting metals and metalloids in samples. It is very reliable and simple to use. It can analyze over 62 elements. It is also measures the concentration of metal in the sample. AAS, or as we called atomic absorption spectroscopy, has an unlimited number of applications and is still a popular choice for uncomplicated trace elemental analysis. FAAS is the widely accepted in many industries, which continue to utilize and unique specific benefits of this technology. Graphite furnace atomic absorption spectrometry, or GFAAS, is an established technology for measuring elements at parts per billion concentrations with incredibly low sample volumes. In the figure of excitation of an atom, the nucleus has electrons leap to higher energy levels, and in the figure 2, which is the relaxation and photon emission of an atom, the nucleus has relaxation and photon emission. Atomic absorption spectroscopy has an instrumentation. First is the hollow cathode lamp. It is the lamp or source of light in AAS. The cathode is made up of a specific element or alloys of elements or coating of element on cathode. When current of 500 volts is applied between anode and cathode, metal atoms emerge from hollow cap and collides with filler gas, which is argon or neon. Due to these collisions, number of metal atoms are excited and emit their characteristic radiation. The second one is burner, with fuel and oxidant. There are different burners are available, which are used to spray the sample solution into fine droplets. Mix with fuel and oxidant so that homogeneous flame of stable intensity is obtained. The most common burners are total consumption burner and laminar flow burner. If the temperature of the flame is too low, it may not cause excitation of neutral atoms. If temperature is too high, it may cause ionization of atoms, and thus sufficient atoms in excited state may not occur. This makes it necessary to select ideal combination of oxidant and fuel, which gives the desired temperature. The third instrument is a chopper. The chopper in the instrument is rotate like a fan. It allows the alternative radiation from flame alone or the radiation from HCl and the flame. This produces a pulsating current or signal, which is used to measure the intensity of light absorbed by elements without the interference by radiation from the flame itself. And then monochromator. Some elements have single absorption line, but several elements have more than one absorption line. Hence, it is necessary to select the spectral line for absorption have measurement. Moreover, it is necessary to isolate the line spectrum of element from that of the emission by the gas in the lamp, or from the background signal of the flame. Hence, a monochromator which can provide good resolution of 1 nm or less is required. For the fifth one, the detector and readout device. It is the intensity of radiation absorbed by elements in UV or visible region from 190 to 780 nm. It can be detected using photometric detectors. The readout device is capable of displaying the absorption spectrum as well as a specified wavelength. The last instrumentation is the interference. Spectral interference, chemical interference, ionic interference, matrix solvent interference dissociation of metal compound. This is the visual representation of atomic absorption spectroscopy. From hollow cathode lens going to the flame, which have a fuel gas and oxidizing gas, nebulizer and the sample solution. From the flame, going to the lens that has monochromator, detector, amplifier, and going to the recorder. Atomic absorption spectroscopy has also a techniques. The first one is the flame atomizer, which frequently abbreviated FAAS. These are the oldest and most commonly used atomizer and atomic absorption spectroscopy. FAAS is most commonly used to toast liquid samples or solid samples which have been dissolved within a liquid. And the second one is the electrothermal atomizer. This atomizer is sometimes also referred as graphite furnace atomizer, as they utilize graphite tube to heat samples rather than a flame. These are the application of atomic absorption spectroscopy. The number one is estimation of trace element in biological fluid like blood, urine, and etc. The number two is estimation of trace elements like copper, nickel, and zinc in food products. The third one is estimation of magnesium and the zinc in blood. The fourth one is estimation of zinc in insulin solution. The fifth one is estimation of mercury in thiomersal solution. The sixth one is estimation of lead in calcium carbonate and petrol. The seventh one, or the last one, is the estimation of elements in soil samples, water supply, effluents, ceramics, and etc. For the addition of application of AAS, it has agriculture and forensic. Agriculture is an atomic absorption spectroscopy as well as atomic fluorescence spectroscopy, which analyzes the light emitted from a sample rather than the light absorbed. These are frequently used in various fields of agricultural study. Typically, they are used to identify and analyze the presence of potentially harmful elements in the environment. The last one is forensic. It is the atomic absorption spectroscopy that has been utilized in the study of forensic sciences for many years. Using this technology, forensic scientists can perform in-depth analysis of blood samples, brain and muscle tissue, and gunshot powder residue.